All right, so this next section of notes is on mutations. And some of the key learnings we want in this first part are to actually figure out what exactly are mutations. And with that, we want to understand that mutations are heritable changes in genetic information. In other words, these are things that can get passed on from one generation to the next. So, when we talk about mutations, we have to understand that one of the reasons why they occur is that because now and then cells actually make mistakes in copying their own DNA, like when they're getting ready to divide. They can insert the wrong base or even skip a base as it is getting put together. And this is very, very common, actually. These variations are called mutations. And this word mutation comes from the Latin word mutare, which means to change. So, like I said earlier, mutations we have to understand are heritable, in other words, inheritable changes in the genetic information. So they are things that will get passed from one generation to the next. Now, if we take all of the possible mutations that are out there, there's two basic categories of them. The first one is those that produce single changes, or produce changes in a single gene, and so these are called gene mutations. And then there are also those that produce changes in the entire chromosome, and those are known as chromosomal mutations. So we're going to go through the different types of gene mutations and also talk about some of the different types of chromosomal mutations as well. So gene mutations. If a mutation within a gene only involves one or even just a few nucleotides, we call that a point mutation. And that's basically because we can almost put our finger to a certain extent on the DNA sequence and say right there, that's where the change takes place. And there are some actually serious diseases where it's just a single base that causes a problem. Something like Tay-Sachs disease, where you have a protein that literally crushes the cells of the brain, and the child usually dies before the age of 5. These typically occur during replication. So again, when we are copying the gene. If a gene in one cell is altered, then that alteration can be passed to every cell that develops from the original one. So to a certain extent, how this affects the individual and how this affects maybe their offspring will depend on how early this takes place in their lifespan. If, this happens in your finger, it may not be a big deal. But if it happens in the zygote, right when sperm and egg come together to create fertilization, every single cell in the body would be affected. So the effects of this has a very, very wide spectrum. Point mutations include 3 different types: substitutions, insertions, and deletions. And we're going to go through those together. So in substitution, it works very much like substituting on a team. You take one base out and you change it into another. So the substitutions usually affect no more than a single amino acid, and sometimes it ends up not having an effect at all. Now, if we look at the example here, we are actually replacing this base here, this cytosine, with thymine. So you can see the cytosine is getting kicked out and the thymine is going in. So that is actually going to change our codon from CGU to CAU, and in this case it does change the amino acid. Sometimes, because we have multiple codons for single amino acids, in most cases, it could potentially not affect it at all, just depending on the substitution. Insertions and deletions we kind of talk about together because they're basically opposites of each other. So they are point mutations where we either insert an extra base or we remove a base from the DNA sequence. So if we add it or delete it, we still read these in groups of 3. But those groupings now shift so that every codon that follows the mutation is basically one off. And so for this reason, we also call them frame shift mutations because they actually shift where we read the genetic message. So if you look here down in the insertion, we inserted the extra A, and you can see it changed all of these amino acids to different ones, and we have an extra base left over, and that doesn't translate into anything. With the deletion, it's kind of the same thing. We remove that G, and so we have two new amino acids, but we're actually short an amino acid here because we don't have a third base to read in the codon. So they can change every single amino acid. That's why frameshift mutations are actually more dangerous than, let's say, a substitution. It can alter a protein so much that it's unable to perform its functions, which means that maybe a process wouldn't happen, or for some other, or some other dire consequence. Maybe an enzyme doesn't work, and you can't digest certain things. Now, chromosomal mutations. This actually involves the overall structure of the chromosome itself. It can change the location of genes. It can even change the number of copies of some genes. So we can create extra copies of genes that we don't need in there. All right, so the first one we're going to look at is deletion. So in deletion, we remove all or part of a chromosome. And so what we can see here is our original chromosome, A, B, C, D, F, and what happens is we delete gene B. And so now it's gone. So we can't actually create that trait anymore. Deletions can be whole genes, like in this case, or it can be part of a gene. It doesn't have to be a whole gene, though. Duplication actually produces an extra copy of either all or even part of a chromosome. So in this case, we duplicated gene B, so we now have 2 copies of it. And this, believe it or not, is a problem. We shouldn't have 2 copies of it. And so if my DNA wants to make actually gene C, it's going to that location and it's thinking it's gene C and it's making gene B again. So it does create a problem. Inversion, what we do is we change the direction of parts of the chromosome. So here we have flipped gene B and gene C, and again, that's a problem because your cell goes to a specific location anticipating that gene B is going to be there, and it's not, it's gene C. Translocation. Translocation occurs when you have another chromosome part that comes and it actually attaches to a different chromosome. So here we have our original chromosome, and here are genes G and H, and they have broken off from their chromosome, and they then go and insert or attach themselves into this chromosome. So it's not even that you're just moving the genes, because you are, but you're actually moving them to a completely different chromosome, which makes it really, really difficult for the body to figure out where it's supposed to be to make that protein. All right, so now that we understand what some of these mutations are, let's start taking a look at some of the effects. How do mutations affect genes? And what we have to understand is the effects of mutations on genes can vary widely. Some have little or no effect, and some produce very beneficial variations. Some can negatively disrupt gene function. Mutations often produce proteins with new or altered functions that can be useful to organisms or changing environments. So when I have a mutation, it can be altered either by a natural event, like some of those chromosomal ones typically are natural events, but there can also be artificial means that actually force this to happen. Now, the resulting mutations can or cannot affect the organism, and it's kind of a genetic roll of the dice. We don't know what those effects are going to be until we actually see the organism and how it functions in the environment. Some mutations that affect an individual organism can also infect the entire species or even the entire ecosystem. As we've learned, many of these mutations are produced by errors in the genetic processes like transcription or even replication. So like I said, some of the point mutations are caused by errors in DNA replication. Now, what you have to understand is this does happen pretty frequently, but look how inserting one base roughly once in every 10 million bases. So that's a pretty good percentage. If you missed 1 out of 10 million points on a test, you'd be pretty darn happy. Now, but these small changes gradually accumulate over time. So as you move along and more replication is taking place, you accumulate more and more errors. And that's where we see these effects. So stressful environmental conditions can also cause some bacteria to increase mutation rates. And I will actually provide you guys with a little bit of a link to a video about Harvard in terms of creating some of these mutations. These mutations can be helpful to the organism, and that's what you will see in the video from Harvard, is that it's actually allowing the bacteria to survive in conditions where it probably should not survive. It's one of the things that drives evolution. So consuming a new food source, resisting a poison. In the case of the Harvard video, it actually does show you how these bacteria are able to live in areas where we have increasing concentrations of antibacterial poison, basically, that they are exposed to. So some mutations, though, arise from mutagens. These are chemical or physical agents in the environment. So these are actually actual things that actually will force mutations to happen. So certain chemical mutagens include things like pesticides, a few natural plant alkaloids, tobacco smoke, environmental pollutants, all of those types of things. Physical mutagens include things like X-rays and ultraviolet lights from the electromagnetic spectrum. We know that, for instance, ultraviolet light causes mutations on our skin. So if these mutagens interact with the DNA, they can actually produce a lot of more mutations that we would get naturally. So some of these compounds will actually interfere with the base pairing, which means more mistakes during DNA replication. Others will actually weaken our DNA strand, causing them to break, which is bad, or inversions, or even translocations on chromosomes. Sometimes the cells are able to repair the damage. There are some cross-checking and different things that take place. But when they can't, the DNA base sequence is actually changed permanently. And once that is changed permanently, then it becomes a question of, okay, is this a helpful or harmful thing for the organism? So the effects of mutations can vary widely. Some have little or no effect. Some produce beneficial variations, whereas some negatively disrupt gene function. So it depends, and this is where I say it's a little bit of a roll of the dice. It is going to depend on how the DNA changes relative to the organism's situation. In other words, the environment comes into play, which sounds like it shouldn't be fair, and unfortunately it's not. It's that combination of the genes that are present and the environment in which they live to see if they work together. We typically think of mutations as negative because they disrupt the normal functions of genes, but it's not always that way. We need mutations in order for evolution to take place. And again, I'll refer you to the Harvard bacteria growth video, because that's what's taking place. Those bacteria are evolving. We need this evolution, and we need this mutations because we need the variation, the genetic variability in our species. Now, if a mutation is harmful, it's because we're changing the protein structure of what is made, or we're changing the gene activity. The defective proteins produced by these harmful mutations can disrupt biological activities and result in things like genetic disorders and genetic diseases, some of which aren't very pleasant and some of which can lead to things like death. I point out cancer. Cancer is the product of mutations that cause uncontrolled growth and division of the cells. But there are some that are beneficial. Some of the variation that is produced by mutation actually is advantageous to the organism or even to the species. Mutations can produce proteins that are new or have altered functions, and depending on, again, the situation, these can actually be useful. It provides a competitive advantage for the organism, which allows them to better survive in their environment. So mutations have helped many insects resist chemical pesticides. Mutations have helped bacteria resist things like antibiotics or antibacterial means that we have to get rid of them. That's one of the reasons why we're kind of in trouble, is we've overused those things. So some mutations have also enabled microorganisms like bacteria to adapt to new chemicals in their environment. So we kind of talked about some of the bad things, but there are some good things. We make use of good mutations. Our food supply actually is very reliant on the use of these good mutations. what we do is we force the chromosomes to not separate during mitosis, and the gametes, when we then reproduce, we can get triploid, which is 3N, or tetraploid organisms. This is a condition that is called polyploidy. And we use this mostly with plants, not so much with animals, but with plants. And like I said, this has affected our food supply because the polyploid plants that we create are larger and they're often stronger than their normal diploid cousins. So, you can have a corn plant that is diploid and a corn plant that is tetraploid. The tetraploid is going to be larger, stronger, better able to resist things like drought or maybe lack of nutrients, and also produce larger amounts of food. There's also some important crop plants like bananas and even limes we've produced this way. Polyploidy is also a natural thing that occurs in citrus plants, so things like lemons, grapefruit, oranges, and these are through spontaneous mutations. So this is how we get some of the different types of, let's say, oranges we have, like navel oranges versus honey bells versus, whatever type of orange you're looking at. Okay, so that wraps up our discussion here on mutations.